Thank you very much, and thank you to the organization for asking me to speak. If you can't see my slides or hear me properly, please let me know. I have my only disclosures are grants and for research and some volunteering positions. Michael Dewan's paper the other year did a very nice job of highlighting where most of the new pediatric hydrocephalus cases come from in the world, and it's heavily dominated in developing countries. The largest number of cases comes from sub-Saharan Africa, but very different from what we see in the industrialized countries are that there's a domination of these cases is coming from postinfectious hydrocephalus, which I abbreviate with PIH here. I have collaborated for a number of years now with the Cure Children's Hospital of Uganda. They've treated over 13,000 cases of pediatric hydrocephalus, but more than 8,000 of these cases are post-infectious. And rather amazingly, almost all of them fail to recover an organism on culture. My first visit there was when Ben Worf was the medical director, and I was fishing for something useful to do, and Ben suggested, well, why don't you figure out what makes these kids sick? And I thought, how hard can that be? So today, I hope to actually show you that we've finally made some substantial progress on this issue. First of all, many of you may have seen this syndrome. It's a young infant who's born normally, gets an episode of neonatal sepsis in the first few weeks of life. The head begins to grow, and on endoscopy, you'll see this pattern of very small pustules lining the ependema of the third ventricle. They tend to be concentrated in the lateral ventricles, actually on the choroid plexus, and we're beginning to investigate that. Amazingly, when you come through the floor of the third, the great vessels and the arachnoid spaces seem never to have experienced an inflammatory reaction typical of a prior meningitis. So these infants often present with a primary ventriculitis inside of the brain without a meningitis. In addition to seeing this in Africa, we're now working in Vietnam. We see very similar cases there. We see it in Haiti and the Caribbean. This syndrome is seen globally in regions that are impoverished. So it's a talk about hydrocephalus, but the pool of patients that generate most of this are children with neonatal sepsis. Neonatal sepsis still takes the lives of about 1 million infants a year on Earth, and the majority of those deaths occur in sub-Saharan Africa and Southern Asia. Substantially more infants that lose their lives than die of malaria, tuberculosis. And we have what I call a microbial dark matter problem here. We don't know the vast majority of the organisms that cause all of these infant infections, even if they don't create hydrocephalus. Very large and well-done studies in recent years looking at the blood of infants with neonatal sepsis. The Cambodian and the South Asian study demonstrated that maybe we recover 2% of the putative pathogens with current technology. The United Kingdom wasn't doing terribly well either in terms of recovering organisms. If we look at spinal fluid, whether in the US or in the Middle East, these numbers are not any better. I just published a review on this in Emerging Infectious Diseases, if you're interested. So it's a massive problem. Our break for this came from the director of the US National Institutes of Health. And with substantial funding to try to crack some of this problem open, we instituted very high quality sampling of blood and spinal fluid in hydrocephalic infants. In regions in Uganda where we didn't have good electricity, we worked with other referring hospitals as well. I won't present the neonatal sepsis cases, but we use liquid nitrogen to do cryogenics. We do have a freezer now at the Cure Hospital. And we put an infrastructure together to get these frozen samples shipped to us in the United States without thawing. And the first question one might throw at this is, what are all of the potential bacterial species that are present in the infants? We look at bacterial genes that are not present in humans when we can't grow an organism. And we didn't leave any chance of failure here. So we split our samples with duplicate samples between a very well-known group at Columbia University led by Ian Lipkin, and then our own genome center led by Jim Broach at Penn State. Very surprisingly to us, we ended up finding that the organism that dominated these infections was a very rare organism. It's called a Painibacillus, and it was a consistent finding in the majority of the cases with hydrocephalus. Here's a composite of all of the CT scans of the first 100 patients. We used case controls, same age. All of these infants are under 3 months of age. We hoped we could capture some of the evidence of a residual neonatal infection, and this was a good decision on our part. But our case controls are congenital hydrocephalics, down below, which also included intraventricular hemorrhage and arachnoid cysts, as you've been hearing in this session. Up here on top, we have two groups of patients. We have diagnostic patients who had Painibacillus recovered, and then there's a group of non-diagnostic ones, and I'll have to talk about these more in the future. As we continue to analyze not just this, but another 300 of these patients. If you see in some of the non-diagnostic infectious patients, there are these small calcified abscesses in the parenchyma of the brain. But in the ones with evidence of Painibacillus, you'll see multiple abscesses and calcified loculated compartments within the ventricular system within these patients. So the brain appearance of these cases really looks different from the ones that are non-diagnostic. We have a marvelous director of microbiology at Penn State, and I asked David Kraft to just throw the book at these, everything he could possibly think of in his long experience in clinical microbiology. So each of the 100 patients had 6 different culture media attempts at recovering organisms. And one of the problems was this appeared to be a facultative anaerobe. So our best means of recovery was actually placing CSF into lytic anaerobic pediatric blood culture bottles. Indeed, although it's a gram-positive organism, they stain gram-negative, which is apparently common in Painibacillus. And this is a species called Thiaminolyticus, which we know fairly well. It's been recognized for about 70 years, but it has multiple insertions that the known reference strains that we've studied, which came from Asia, don't possess. And so we named this strain after we assembled its genome and named the strain after the city where it was discovered, Mbali. A very great group of young folks at Penn State, and Christine Henley and Patty Santango then took the reference strain that we have for this organism and put it into littermates of mice, along with the same colony count of the African strain. The reference strain indeed is a very benign organism, The African strain is almost universally lethal when given to mice. We're in the process right now of trying to figure out just how it kills these animals. There's tingible body necrosis in the spleen and acute tubular necrosis in the kidney from what looks like toxins. And we're using CRISPR editing to try to clip out the toxin candidates that seem the worst in this organism. And the next question you might ask if you had enough resources is, well, why would there only be one organism doing this in these infants? So we use multiple modern techniques. This is a capture technique here. We use deep RNA sequencing separately to look at the products of viruses. And we ended up finding that in about a third of these infants, they had cytomegalovirus, active cytomegalovirus infection as well. The only time we saw that in the CSF was in the postinfectious hydrocephalic cases, not in the case controls. So I now coded here with red stars for blood and yellow stars for CSF, the cases who had active cytomegalovirus infection. We still don't know the role of this, but congenital hydrocephalus and early perinatal, congenital cytomegalovirus infection is quite dangerous to children. It affects their neurological outcome. We typically treat such children with antivirals in the US and in Europe. Those medication will induce neutropenia, and we've not been willing to introduce clinical trial yet, testing of such medication in developing countries. All of this just came out in Science Translational Medicine. And what I've just shown you doesn't scale. What do I mean by that? Well, you can't take several $1 million worth of funding and investigate over 4 or 5 years every pediatric neurosurgical site throughout the world if they need this type of analysis done. So how can you approach this if you only have a little bit of surveillance? We're taking all the villages in Uganda. There's about 50,000 now. And we're mapping them onto these satellite grids that we get our weather and environmental information from. We published a number of years ago that these cases come in with the rainy seasons. And so there's an environmental factor which is important and which we'd like to track down with the Pediatric cases. Indeed, if you look at where they're from, these are control cases spread throughout the population centers of Uganda. The infectious cases are much more concentrated on the north and south banks of a large lake that the Nile River flows into. It's a swampy region. In fact, almost all the Pediatric cases are very tightly clustered. So this looks from its genomics and from its location as a likely soil or waterborne bacteria. And we look forward in the next year to doing the field work needed. We've been a bit disrupted by the pandemic, as has everyone. But the best surgical options aren't good enough for this. And I think we tend to get tunnel vision as surgeons. And we've looked, along with Abigail Carney and Ben Worf and another very good team of people, at brain growth in these infants. They tend to come in with small brains. About a quarter of them will have some catch-up growth in the first year of life. This paper was in the New England Journal a few years back, but we're just now processing the longer-term outcome. And it looks like the results will be disappointing. These infants got catch-up growth early in life, but they're not continuing to grow, their brains are not growing adequately, nor is their cognitive ability improving along the lines that any of us clinically would be satisfied with. So I'll leave you with some thoughts. I think it's important that we avoid what I call surgical tunnel vision. We think that surgical excellence is enough, but we can't operate our way out of epidemics and public health disasters. And I think what we're dealing with in East Africa with these hydrocephalic infants is an epidemic, an infectious disease epidemic. We also can't fix their heavily damaged brains. So we're palliating these infants. And on the other hand, nobody's going to help us. We're the experts for neurosurgical disease. And I think that it puts us in the uncomfortable position of having to lead the type of infectious disease analysis and public health measures needed to control our own cases. I think that global neurosurgery for cases like this really have to integrate into efforts at sustainable global health. And also that what we're showing is that most of the global burden of hydrocephalus in countries such as this are preventable. So we only can do that collaboratively. And everything I showed you today was the very, very hard-won effort of many, many people in Africa, Europe, and the US and Asia to try to address these large numbers of cases that are seen around the world. And I thank you so much for your attention today.
Thank you, Steve. That's a fantastic talk and much food for thought. I have a question from one of the other panelists, Anthony Figaji, has asked, from what you've said in the presentation, the samples were collected entirely in Uganda, but the talk title suggested a larger study in Africa. And given that Africa is a diverse collection of 54 countries with great differences between them economically, geographically, and probably bacteriologically, how is it possible to generalize these data to the whole continent as the title suggests? And I would like to comment on that.
That's exactly some of the key issues I think that we face. So far as I know today, the importance is not the particular organisms that we found in this particular place, but the techniques that we used, which is an unbiased screening for the presence of non-human pathogens that are present in the brains of infants, and they can give you enough inflammation to generate post-infectious hydrocephalus. Now, the technology to do that today tends to be expensive, and doing it in the way we've done it, concentrated in large centers that are far from the impoverished areas where it needs to be done. But we can make this a much more sustainable approach, first of all. And there are technologies that usually fall under the category of nanopore sequencing that can do an unbiased approach with a device you can hold in the palm of your hand about the size of a cell phone. But we don't have the workflows yet, and we really need more robust systems for that. The second is we need to survey the other places where the majority of these cases are. We may find that Pediatric is present in many of these places, and we may find, which is my fear, that we're going to have to customize how we approach, treat, and prevent these illnesses in many different parts of the world that, as you asked, will indeed show unique organisms.
Thank you. I have some questions coming through live. First one from Adrian Casersis. What is the ideal growth medium for Pediatric, and how hard is it to grow for us with less sophisticated technology at our disposal?
This week, we're actually trying to make good use of more pediatric anaerobic lytic blood culture bottles. I'm saying all of those components because this organism may be intracellular in children with high white counts in the CSF, and therefore breaking up the white blood cells that contain the organism may be as important as the anaerobic conditions. Once we've passaged this organism anaerobically, it will grow well on McConkey's agar in the lab in the presence of oxygen. But those are all of my suspicions, as I'm now still using this week to try to recover more variations of the species.
And a second question from the webinar viewers from Dr. Okuyo. What is the antibiotic treatment that is used for pain bacillus, or is there an antibiotic therapy for it?
Yes, so far, this seems to be a very sensitive organism. And I say so far because if I can get more than just 3 painibacilli cultures, but I have 24 cultures, then for the full spectrum of antimicrobial resistance and sensitivity may be different. But so far, this looks like a very sensitive organism to all the common antibiotics. The problems are pretty obvious. First of all, in calcified abscesses in newborns, we have a penetrance problem to get it in. We're going to have to be using surgical drainage if we're unable to get good penetrance to kill this organism. And it needs to be treated early enough in the course of an infection where with negative cultures, we really are blind to the progress of treating the infection. So we're putting into place PCR reaction technology at the Cure Hospital in Uganda so that we know who has this organism, and also critically, we know how long to treat with antibiotics to clear the CSF before we wander in and do a shunt or an ETV in an actively infected child.